Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. We will now go to lecture number 48, where I will speak about the origin of phenolics via shikimate pathway. So, phenolic compounds are very important because they plays important role, very important role. So, I will talk about that, but let me briefly tell what are the concepts to be covered in this lecture. So, as you see here that we will first cover the shikimate chorismate pathway, and then we will talk about the regulation of shikimate pathway, and then we will talk about the biosynthesis of aromatic amino acids. Okay. So, phenolics, as we say, so phenolics is basically a phenol containing a hydroxy group, we will come to that. So, why we study phenolics and phenyl propanoids? So, phenolics is a big name. So, it starts with mostly it starts with phenyl propanoids. So, phenyl propanoids are generally C6C3 compounds. So, right, for example, this is C6 and this is C3. So, this is a 3 carbon and this is a 6 carbon. So, these are phenyl propanoids. So, why we study phenyl propanoids? Because phenyl propanoids consist of diverse subgroups like coumarins, like lignans, like lignins, like suberin, stilbene, flavonoids, tannins. So, most of this we are going to cover in the subsequent class in the lecture number 49. Okay, let us see. So, this is phenol. Phenol is just a simple benzene ring with a hydroxy group. So, any compound containing a benzene ring with a hydroxy group will come under phenolics. But again there are compounds where hydroxy group is not there, but still we put it under phenolics. Classic example is this is a cinnamic acid. So, here there is no hydroxy group, but still we will call it phenolic compound because this is the origin of phenolic moieties. So, here from where it starts from phenylalanine it converted into cinnamic acid. So, you can say this is the basic precursor of the phenolic compounds, and apart from that there are a lot of phenylalanine derivative phenolic compounds are there which contributes to the rose fragrance like phenyl ethanol, then phenyl ethyl acetate. So, where also there is at 4th position no hydroxy is there, but still we will call under phenolic compounds. So, what we are going to see that how phenolic compounds originate. So, it basically originates from primary metabolism, as you see here erythrose phosphate and phosphoenol pyruvate, these two joins and eventually they create the shikimate pathway, which where shikimate is one of the major intermediates, and then it produces chorismate. So, sometimes we call it shikimate chorismate pathway. From chorismate pathway there are two different routes. One route contributes to the tryptophan. So, we have studied the role of tryptophan particularly in case of indole alkaloids. So, where tryptophan converted into tryptamine by tryptophan decarboxylase, and we have also said how tryptophan originates via indole. And the other route which is of much concern in these classes is basically the formation of tyrosine and phenylalanine. So, tyrosine and phenylalanine, these tryptophan, these three are aromatic amino acids, and the difference between tyrosine and phenylalanine is this, that in case of phenylalanine, This is the structure of phenylalanine, and when this 4 position OH is added, then it becomes tyrosine. So, let us now see in details about the shikimate chorismate pathway. The first step is basically erythrose 4-phosphate joining with phosphoenol pyruvate here, and the E1 what I have mentioned here is basically the first enzyme which is called DAHP synthase. DAHP synthase. Also nowadays it is called DHS as well, DHS. It is also called DHS as another abbreviations. So, this is 3-deoxy-D-arabino. So, let me write it for you, 3-deoxy-D-arabino heptulosonic acid 7-phosphate, which is called DAHP. Okay. So, this is DAHP. This DAHP in the next step it is converted into 3-dehydroquinate. So, the 3-dehydroquinate, the enzyme which is E2. E2 is basically dehydroquinate synthase. Dehydroquinate synthase we also call DHQS. Next enzyme is E3. E3 basically dehydroquinate converted into dehydroshikimate. Dehydroquinate converted into dehydroshikimate, so or dehydroshikimic acid. So, the enzyme is dehydroquinate, DHQ stands for dehydroquinate, and then shikimate, shikimate dehydratase. Dehydratase means basically a molecule of water will go out. So, you can show it here H2O. So, this forms dehydroshikimate. So, the next one is the E4. E4 is also, this is dehydroquinate, shikimate dehydrogenase. Shikimate dehydrogenase. So, that is called DHQ dash SDH, dehydroquinate shikimate dehydrogenase. Okay. Here the abbreviations is also same for the other one. So, here also DHQ SDH, abbreviation is same for both the cases, whereas the first one is a dehydratase and second one is the dehydrogenase. So, when is a dehydrogenase, then it requires a NADPH. And liberates NADP plus. Okay. So, it forms shikimic acid, which I mentioned that this is one of the intermediate, major intermediate, that is why the pathway is named as shikimic acid. Next what happens, shikimate will be converted into shikimate 3-phosphate. So, there will be the addition of phosphate, that means ATP is required. So, you can add an ATP, it liberates ADP, which forms shikimate 3-phosphate, and this shikimate 3-phosphate, then what is the enzyme? Enzyme is basically it is as phosphate, this is called shikimate kinase. Shikimate kinase SK. Okay. And the next one, shikimate 3 phosphate with addition of phosphoenol pyruvate, it formed EPSP. So, EPSP is again another major product of this pathway, EPSP, and the enzyme E6 is called EPSP synthase. So, and then E7, which converts EPSP to chorismate. So, the E7 is basically it's CS, we can say it is chorismate synthase, CHORISMATE chorismate synthase or CS, and this one is PS. Okay, here one important thing I would like to tell here, that one of the very well known herbicide is glyphosate. So, glyphosate is applied to get rid of the weeds. So, basically it stops the shikimate pathway, and that thus blocks the formation of aromatic amino acids, eventually the metabolism, and the plant dies, particularly the dicots. And what it happens, that glyphosate basically blocks here. So, it is basically a competitive inhibitor of this enzyme, which is EPSP synthase, and therefore, the pathway blocks. So, if one study the pathway leading to formation of the different phenolic compounds, if metabolism can be studied in cell and organ culture, there glyphosate addition can make impact on the end product. So, regulation is so stringent. So, this is in brief about the pathway leading to the formation of chorismate from erythrose phosphate and dihydroxyacetone phosphate. Next slide, what we will see, that chorismate, what we see, that will be converted into this is called prephenate. So, the prephenate is basically called chorismate mutase. So, this enzyme, so I will call CM. CM stands for chorismate mutase. Then comes this prephenate. So, prephenate is here. Prephenate will be converted into L-arogenate by another enzyme, which is called prephenate aminotransferase. Chorismic acid prephenate to prephenate will be converted, yeah, it is called prephenate aminotransferase. We will also call this in brief as PAT. The enzyme is called PAT, E9, what is the new name is PAT. So, PAT means that prephenate aminotransferase. So, it is written here prephenate aminotransferase. Okay. And next what happens, one arogenic acid is formed. So, but this requires glutamate, and which liberate 2 oxoglutarate. Okay. And then it converted into arogenate. So, arogenate, from there are 2 pathways exist. One is arogenate dehydratase. So, ADT. So, I can give a name ADT here, this one is ADT. ADT stands for arogenate dehydratase. So, arogenate dehydratase. When we say so, then we have to show that a water molecule is released as well as a molecule of carbon dioxide is also released. So, which converts arogenate to tyrosine. The other one is that arogenate dehydrogenase, which is ADH, which is aragonate dehydrogenase. When it requires, again it here also a molecule of carbon dioxide liberates, and it requires this NADPH. So, because of shortage of space I am not showing that, and it finally, that is why ultimately it makes phenylalanine or tyrosine. If aragonate dehydrogenase work, it makes phenylalanine. If aragonate dehydratase work, it makes tyrosine. And in plants both the pathways operating, but there is a fine balance between two, and we will see much later that the volatile benzenoids originate from the phenylpropanoids. So, those and they emit from different flowers. Petunia was used as a model. So, scientists what they did, they tried to control the pathway at this level, ADH and ADT, and that basically makes impact on the final product. So, that means the control is far ahead than the, it is not in the penultimate step, but in the very long distal step ahead. Now, what I will say that subcellular localization. So, the pathway mostly operate in the plastid. So, that means the choriamic acid onwards the pathway is operating in the plastid. So, like choriamate mutase, okay, and then this prephenate aminotransferase, then aragonate dehydrogenase, then aragonate dehydratase, both are operating inside the plastid, and tyrosine and phenylalanine are produced inside the plastid. Okay. And from there maybe they are emitting out into the cytosol for different function, whereas cytosolic phenyl pyruvate pathways also exists, okay, where phenylalanine is produced from choriamic acid. So, here you see choriamate mutase 1, this is CM1, which is operated in the plastid, whereas CM2 choriamate mutase 2, which convert choriamic acid to prephenic acid, this is isoform, but this basically localized in the cytosol, which makes prephenic acid. Then there may be a prephenate dehydratase, which is still not clear, but which makes phenyl pyruvate. This phenyl pyruvate, by phenyl pyruvate again aminotransferase, which basically makes the phenylalanine. So, this is the cytosolic route of formation of phenylalanine from the shikimate pathway. Okay. So, now both phenylalanine and tyrosine are formed. So, this is in a nutshell the pathway. Now, what I am showing here is basically this is from a review article which is published in Current Opinion in Plant Biology 2022. So, where basically it focused on that the shikimate choriamate pathway, the main regulation occurs at the very first stage, which is this DAHP synthase, which I have also said that it is nowadays called DHS also. There are different isoforms exist like DHS1, DHS2, DHS3, and that basically plays important role in controlling the pathway. So, basically some feedback regulation work. So, when sufficient amount of tyrosine is produced, then a signal goes and stops the DHS2, the red one. So, which isoform is basically controlling the tyrosine, whereas the even choriamate can also be controlled by that. Even much later the caffeate, which plays important role in lignification and other caffeic acid esters, that is also being controlled at that very early stage in the pathway. So, okay. So, DHS is important, that is why I made it. Otherwise, the whole pathway more or less I have covered. So, only to tell you that the where the regulation lies. And also another important point, this from tyrosine different metabolites originate like tocopherol, vitamin E, plastoquinones, isoquinoline alkaloids, we have covered that. Cyanogenic glycosides we have not covered in that details, betalains. Whereas from phenylalanine, the most important products are lignins, tannins, suberin, flavonoids, anthocyanin, coumarin, stilbene, salicylate. This we are going to cover soon. And tryptophan is also being controlled by the DHS2, and we have seen that tryptophan contributes to indole alkaloid pathway. Now, multiple regulations of plant DHS activity as indicated by the pink box. So, these DHS genes are basically nuclear encoded genes, and then once the DHS protein is produced, it mostly moved into the plastid. Okay. So, basically this is showing the movement into the plastid, and there it basically performs its function, and there manganese ion plays an important role and it performs different functions. So, basically it plays important role in this feedback regulation. This is what I mean to say, and there is also cobalt dependent cytosolic DHS activity has also been discovered in recent years. So, but this is not fully known, and this is again from the latest information available in current opinion in plant biology. So, the take home message is that DHS or DHP synthase is very important enzyme, that is the regulatory enzyme which is controlling the shikimate corismate pathway, ultimately the phenylpropanoid pathway, and that is mostly it moved into the plastid, but there is also the evidence of cytosolic DHS. So, so this is we have discussed the shikimate pathway. So, shikimate pathway forms the phenylalanine, here we have discussed. So, phenylalanine are C6C3 compounds, C6C3 compounds, and from there different other C6C3 compounds originate like cinnamic acid, coumaric acid, coumaryl CoA, and then what happens from this C6C3, then again this is C6C3, and from there it forms lignin, which is basically C6C3, and lignin is multiple N. So, it is a polymer, and it can also form simple phenolics like C6C1 benzoic acid, C6C2 compounds, even the C6 C3 C6 which is called flavonoid compounds, this can also produced, and this when joins in multiple units it forms the condensed tannins of proanthocyanidins. Also from shikimic acid the gallic acid is produced, and which plays important role in forming the large polymer hydrolyzable tannins, which I will show in the next class. So, the point here is this, that shikimate aerogenate pathway ultimately contributes to phenylalanine or hydroxycinnamate pathway. So, phenylalanine to cinnamic acid. So, the cinnamic acid is hydroxycinnamic acid when this cinnamic acid when it is converted into 4-hydroxycinnamic acid, which is called paracoumaric acid, we will see that. So, that is why it is called hydroxycinnamic acids. So, that is an important molecule from where from that molecule different compounds originate. So, if you remember at the origin of when you talk about the origin of specialized metabolites. So, from the precursor it produces the main P product, and from P undergoes different transformations. So, basically these are nothing but different transformation which occurred in different plants. Sometimes it occurred in several of these reactions occurred in different plants. For example, 1 to 5. So, what is showing here 1 to 5, 1, 2, 3, 4, 5. So, 1 to 5. So, this basically leads to formation of flavonoids. So, this is flavonoids, and then number 2 is basically stilbene. We will talk all this in the subsequent class. Okay. Then stylopyrones, we will leave it. Benzophenones, benzophenones actually leads to the formation of this xanthones. This is number 4, is very important. Ultimately, it forms the xanthones, and this actually contributes to anthocyanin. And then what important is this dihydrocinamate, cinnamyl 7 to hydroxycinnamyl alcohol. So, this is basically the component of lignin monomer, which is called monolignol. Then number 8 is basically hydroxybenzoates. These are small molecules, so which includes vanillin also. And then number 9 is basically hydroxycoumarins. So, coumarin you will find in several plants including umbelliferae species, and then conjugations which leads to hydroxycinnamate esters. Number 10 we will see that in due course, and amides. So, basically this is the central role of hydroxycinnamates towards the formation of different phenylpropanate compounds. So, these are the transformation P1, P2, P3, P4, P5. So, P is basically the hydroxycinnamic acid. So, now the major class of phenolics, I will end this class briefly telling this, but again I will bring this issue in the subsequent class. So, C6 are called simple phenols, examples are catechols. C6C1 are the hydroxybenzoates, C6C2 are acetophenons, C6C3 which are phenylpropyens, hydroxycinnamates, coumarins. Then C6C4 are naphthoquinones. We will talk about one such naphthoquinone pigments towards the end of this phenolic classes, seconin. Then C6C1, C6 is basically the xanthone, whereas C6C2, C6 which is stilbene, and the C6, C3, C6 are basically flavonoids, and then C6, C3 constitute the lignin with N, okay, and then so on. Then C6, C3N which is condensed tannins, it is basically the polymer. So, we will talk about these important groups in the subsequent classes, like we will talk about lignans, which is basically 2 molecules of monolignon, lignin monomer joins and form lignans. We will mention about that, and so many interesting things. So, this is basically the major classes of phenolics. All these originate from this shikimate corismate pathway, and these are some of the structures with this. So, like hydroquinone, like catechol, 4-hydroxybenzoate, acetophenone, caffeic acid, eugenol, we will talk about that, and then resveratrol, we will talk about that, tetrahydroxyxanthones, we will talk about that, quercetin, we will briefly talk, pinoresinol, we will talk, and so many, so on. So, with this I end this class. Thank you very much.